Good afternoon, friends. In last lecture, I have covered or I discussed the structure of bacteria. Okay, next part, that is in structure of bacteria, the cell wall is also present. We will see the discussion or description about cell wall. So, what is cell wall? It is rigid structure which gives definite shape to the cell. That is, it means the shape of bacteria is due to the presence of what? That is cell wall. It is rigid structure which gives definite shape to the cell. Then it is what when it is located, it is situated between the capsule and cytoplasmic membrane. Where it is located, or where the cell wall is located, it is situated or located between what? The capsule and cytoplasmic membrane. Then next, it is about 10 to 20 nanometer in thickness, and it constitutes about 20 to 30% of dry weight of cell. Keep in mind the thickness of cell wall, it is 10 to 20 nanometer, and it constitutes of what? That is 20 to 30% of dry weight of cell. Then next, the cell wall, it direct cannot be seen by direct light microscopy, and it does not stain easily by different staining reagents. Next, the cell wall of bacteria, it contains diaminophilic diaminophimalic acid, that is DAP, and muramic acid and teichoic acid. Yes, what it contain? The cell wall contain DAP, that is diaminophenolic acid, then muramic acid and teichoic acid. These are the substances are joined together to give rise to complex polymeric structure, and this structure is known as peptidoglycan or murin or mucopeptide. Yes, what I am saying, I am repeating once again. The cell wall of bacteria, it contain what? DAP, that is diaminopimelic acid, then muramic acid and teichoic acid. And these three substances are joined together to give rise to a complex polymeric structure, and this complex polymeric structure is known as, yes, this known as mucopeptide, or it is known as murin, or it is called as peptidoglycan. Next, the peptidoglycan is the major constituent of cell wall of gram-positive bacteria, and it is 50 to 90%, and in gram-negative bacteria the cell wall is present only at 5 to 10%. Keep in mind the difference between the cell wall of gram-positive and gram-negative bacteria. In gram-positive bacteria, the cell wall, it is 50 to 90%, and in gram-negative bacteria it present only 5 to 10%. Next, the chemical structure of bacterial cell wall, it contains N-acetylmuramic acid, then N-acetylglucosamine, then peptide chain and pentapeptide chain. This is about the chemical structure of what? That is bacterial cell wall. Next, we will see the difference between cell wall of gram-positive bacteria and cell wall of gram-negative bacteria. First, that is thickness. The cell wall, the thickness of cell wall in gram positive, it is 20 to 25 nanometer, and in gram negative bacteria it is 10 to 15 nanometer. Next, peptidoglycan, it is present more, that is 50 to 90%, and it is present in less, that is 5 to 10%. Then teichoic acid, it is present in gram positive bacteria and it is absent in gram negative bacteria. Then polysaccharide, it is present in gram positive bacteria and it is absent in gram negative bacteria. Then lipids, in gram positive bacteria it is less or it is absent. In gram negative bacteria it is present in more quantity. Then cell wall, in gram positive bacteria it is simple cell wall. In gram negative bacteria it is complex. Then the outer membrane and periplasmic space, it is absent in gram positive bacteria, and this outer membrane and periplasmic space is present in gram negative bacteria. Then effect of lysozymes In gram-positive bacteria, it is easily destroyed. And in gram-negative bacteria, it is resistant. Then type of amino acid, it is few, and in gram-negative, it is several. Then susceptibility to the streptomycin and tetracycline. In gram-positive, it is slight, and in gram-negative, it is marked. Then susceptibility to penicillin and sulfonamides. In gram-positive bacteria, it is marked, and in gram-negative bacteria, it is much less. Then example of gram-positive bacteria: Bacillus anthracis, Clostridium tetani, then Staphylococcus aureus, and Corynebacterium diphtheria. Yes, in examples of gram-positive bacteria: Bacillus anthracis, then Clostridium tetani, Staphylococcus aureus, and Corynebacterium diphtheria. Then examples of gram-negative bacteria: E. coli, that is Escherichia coli, then Salmonella typhi, then Vibrio cholerae, and next example Haemophilus influenza. These are the examples of gram-positive bacteria and gram-negative bacteria. And this is the difference between what? Cell wall of gram-positive bacteria and cell wall of gram-negative bacteria. Then we will see the functions of cell wall. First function, see here, what is first? That is cell wall involved in growth and cell division of bacteria. That is cell wall, it plays an important role in what? The growth of bacteria as well as cell division of bacteria. Then next, it is important, or it gives shape to the cell. That is, it is important for, that is, shape of cell. Then next, it gives protection to the internal structure, and it acts as a supporting layer. Or third function, it gives the protection to the internal structure and it acts as a supporting layer. Next, it prevents the rupture of bacteria which is caused by osmotic pressure, that is differences between the intracellular and extracellular environment. That is, next function, it prevents the rupture. Next, it provides support for the flagella. Yes, flagella, it is responsible for what? That is motility or movement. That is function of cell wall is to provide the support for flagella. Next, to regulate the certain degree of passage of molecules into and out of the cell. Next, it serves as a site of attachment. It serves as a site of attachment for most bacterial viruses. These are the functions of what? That is cell wall. Next part, that is ribosomes. Ribosomes are the center of what? That is protein synthesis. They are slightly smaller than the eukaryotic ribosomes. The sedimentation constant is 70S. These 70S ribosomes are made up of two subunits, namely the 5 subunit 50S, and these 70S ribosomes are made up of two subunits, that is first large subunit 50S and small subunit, that is 30S. Here we can see the 30S and 50S ribosomes. Here we can see the image. During active protein synthesis, the ribosomes are associated with mRNA, and such associations are called as polysomes. Here we can see the image of polysomes and also mRNA. What I am saying, during the active protein synthesis, the ribosomes are associated with mRNA, and these associations are called as what? That is polysomes. Here we can see the structure of polysomes, the structure of 30S, 50S ribosomes, and structure of mRNA. Next part of bacteria, that is spores. The process of endospore formation is called as sporulation, In this process of sporulation, it may take 4 to 8 hours in vegetative cells. Yes, what is sporulation? It is the sporulation. It means it is the process of endospore formation. In the process of sporulation, it may take 4 to 8 hours for 4 to 8 hours for sporulation in vegetative cells. Then endospores are thick walled, highly refractile bodies that are produced one per cell. Keep in mind, see here, these spores are produced one per cell. Each bacterial spore on germination, it forms a single vegetative cell. Therefore, this sporulation in bacteria is a method of preservation and not it is a method of reproduction. What I am saying, the each bacterial spore, after their germination, it forms the single vegetative cells, and therefore this sporulation in bacteria, it is the method of what? It is the method of preservation, not for the reproduction. Then spores are extremely resistant to desiccation, then staining, disinfecting the chemicals, radiation and heat. The spores are resistant to what? Heat, then resistant to radiation, resistant to staining, then resistant to disinfectant chemical and resistant to desiccation. Next, they remain viable or centuries, and it helps in bacteria to survive for long period under unfavorable environment. This is so important, that is, it remain viable and it help in bacteria to survive for long period of time under unfavorable environment or unfavorable condition. The endospore can remain dormant for thousands of years. The spores of all medically important bacteria are destroyed by moist heat sterilization, that is by use of autoclave at 121 degrees Celsius for 20 minutes. There are two methods of sterilization. First, that is dry heat sterilization, and second, that is moist heat sterilization. In dry heat sterilization, it means by the use of hot air oven, and in moist heat sterilization by the use of autoclave, at which temperature, that is 121 degrees Celsius for 20 minutes. This is all about the ultrastructure of bacteria. Next, we will see the classification of bacteria. There are a number of ways we can classify the bacteria. First, the based upon several major properties. First, based upon morphological properties, then anatomical, then staining property, then classification based upon pathogenicity, then classification based upon relationship of host and organism, then classification based upon nutrition, and last, classification based upon environmental factors. We will see one by one. First, morphological classification. The bacteria are classified according to the morphological characteristics. The morphological characteristics, for example, shape, size, structure, and their specific arrangement. For example, motility and flagellar arrangement. The bacteria are classified against 6. On the basis of morphology, it is divided into 6. First, true bacteria. In that first, cocci. These are the spherical shape or oval shape cells. On the basis of arrangement of individual organism, they can classified again. First, monococci. It means cocci in singles. For example, monococcus species. Then diplococci. Then cocci are present in pairs. from name itself indicate mono it means what, single, that is cocci in single. Then diplococci, that is cocci in pairs, for example streptococcus pneumoniae. Then staphylococci, that is cocci in grapes, it is grape like structure or cluster. Staphylococcus example, for example Staphylococcus aureus. Then streptococci, that is cocci are present in chain, for example Streptococcus pyogenes. Then tetrad, the pres- cocci is present in group of 4, that is Micrococcus coccus species. Then Sarcina, that is cocci in group of 8. That's what I'm saying. Monococci, it is cocci in single. Cocci are present in single. Then diplococci, cocci are present in pair. Then staphylococci, it is grape-like. Then streptococci, cocci are present in chain. Then tetrad, cocci are present in group of 4. Then next Sarcina, cocci are present in group of 8. Next, bacilli. These are the rod-shaped bacteria. On the basis of arrangement of organism, they are again classified as a diplobacilli, then streptobacilli, palisades, Chinese letter form, cocobacilli, and comma-shaped. Then next, actinomycetes. Actin, it means rage, and mycase, it means fungus. These are the rigid organisms like true bacteria, but they resembles fungi in that they exhibit the branching and tend to form the filament. Then they term because they resembles to sun rays and seen in the tissue section section. Then spirochetes, these are relatively longer, slender and non-branched microorganisms, this having spiral shape and having several coils. These are spiral shape back and it having the several coils. Next, mycoplasm. These are the bacteria lacking rigid cell wall, that is cell wall lacking, and highly pleomorphic and indefinite shape. Keep in mind here it is important. What is mycoplasm? These are the bacteria lacking rigid cell wall, that is cell wall lacking, and it is highly pleomorphic and indefinite shape. Then they occur in round or oval bodies and it is in interlacing filaments. Then rickettsia and chalamate, these are the very small obligates parasites, and one time where it is considered closely related to the viruses, now these are regarded as a bacteria. Next, classification based upon anatomical features. First, that is capsule. It means these are the bacteria which shows presence of capsule, that is capsulated. For example, Streptococcus pneumoniae. The next, non-capsulated, that is these are the bacteria which shows absence of capsule, that is for example Viridian streptococci. Next, flagella. It means this classification is based upon flagella, presence or absence of flagella. For example, it is classified monotrichous, lophotrichous, amphitrichous, and peritrichous. I have already discussed in last lecture, that is monotrichous, what is monotrichous, lophotrichous, what is amphitrichous, and what is peritrichous. Then next, a flagellate example, Shigella species. Next, spore. First, The spores forming, that is Bacillus species, and non-spore or non-sporing or non-spore forming, that is Escherichia coli, that is E. coli. Then classification based upon staining reaction. First, that is gram stain. In gram staining, first gram positive bacteria and second gram negative bacteria. In gram positive, these are the bacteria after the gram staining which shows violet in color, and in gram negative bacteria after this gram staining or after the staining it shows pink or red color. This is difference between gram staining, gram positive and gram negative. This gram positive bacteria it shows which color, that is violet color. And second, in gram negative bacteria, this gram negative bacteria which shows presence of what, that is pink or red color. Next, acid fast stain. In that, first acid fast organism and second non-acid fast organism. In acid fast organism, after the staining, the Zell-Nielsen reagent or stain, it will shows the pink color bacteria. In non-acid fast, after the staining, it shows blue in color. So what is the difference between acid fast organism and non-acid fast? That is non-acid fast organism, that is the acid fast organism, it shows presence of which color, it shows presence of pink color after the staining. And non-acid fast organism, it shows blue color after the staining. Next, gram stain and acid fast stain. Gram positive cocci, example Staphylococcus aureus. Then gram negative cocci, Neisseria gonorrhoeae. Then gram positive rods, it is Clostridium species, and gram positive rods, Clostridium species, and gram negative rods, it is E. coli, that is Escherichia coli. Then acid fast stain. In acid fast bacilli, Mycobacterium tuberculosis, and non-acid fast bacilli, it is Staphylococcus aureus. Then next classification based upon pathogenicity. First, pathogen, the organism which is able to spread disease and can cause the disease. The non-pathogen which does not causes any disease. For example, lactobacilli. And next, commensals, normally non-pathogenic. These are normally non-pathogenic. It means it does not causes the disease, but sometimes they shows disease when immunity impaired. Keep in mind the commensal, these are basically what, non-pathogenic. Non-pathogenic, it means it does not causes the disease, but sometimes they shows the disease when immunity is impaired. Next classification based upon the relationship of host and organism. First, spirophytes. These are the free living microbes on dead animals or decaying things that can be found in soil and water, and it plays important in degradation of organic matter. What is spirophytes? That is, these are the free living microbes which are present or which are on dead animal or decaying things that can be found in soil and water, and it is important in the degradation of organic matter. Next, parasite. These that are established themselves and multiplies in hosts and multiply in hosts. Then commensals, these are the microbes that live in complete life with harmony, but they can produce diseases when host response is lowered. Example, normal flora organism. Then next is based upon nutrition. autotrophs and heterotrophs. Then next classification based upon environmental factor, for example temperature, salt concentration, pH, then oxygen dependence and atmospheric pressure. First temperature. First psychrophiles, for example Pseudomonas fluorescens. Then mesophiles, Escherichia coli, then Salmonella enterica and Staphylococcus aureus. Then thermophiles, it means 52 degrees Celsius, 50 to 60. For example, Bacillus Stereothermophilus. Here we can see the temperature of each and every category. And extremely thermophiles, it means at as high as 20 to 50 degrees Celsius. Next, oxygen dependence. First, aerobes, that is which grows in ambient temperature, which contain 21% of O2 and small amount of CO2. That is aerobes, it having which contain the 21% of what, that is oxygen, and 0.03% of CO2, that is carbon dioxide. Then obligate aerobes, it strictly requires O2 for their growth and that is oxygen for their growth. For example, Pseudomonas aeruginosa. Then microaerophilic, these are the bacteria which grows under reduced O2, that is 5 to 10%, and increased CO2, that is 8 to 10%. For example, Campylobacter jejuni and Helicobacter pylori. These are the examples of what? Microaerophilic. It means it grows under reduced O2, that is 5 to 10%. Increased CO2, that is 8 to 10%. Then next factor, that is pH. First, acidophiles, example Lactobacillus acidophilus. Then alkaliphiles, that is Vibrio. Then neutrophiles, that is having pH 6 to 8. The majority of the medical important bacteria which grows best at neutral or slightly alkaline pH or slightly alkaline reaction, that is having pH 7.2 to 7.6. Then other way of classification, motile or non-motile, then pathogenic or non-pathogenic, then sensitive or resistant bacteria, sensitive or resistant to what, that is particular antibiotic or any particular chemical. Then lactose fermenter or lactose non-fermenter. What are the other ways of classification? First, what I am saying, the motile or non-motile. Second, pathogenic or non-pathogenic. Then sensitive or resistance. For example, sensitive or resistance to any particular antibiotic or any particular what, chemical. Then lactose fermenter or lactose non-fermenter. Then classification based upon cultural characteristics. For example, growth rate. First, rapid grower, for example Vibrio cholerae, and slow grower, for example Mycobacterium tuberculosis. Then next pigment production. Then pigment producer, for example Staphylococcus aureus, and pigment non producer, that is Escherichia coli. Classification based upon what? Cultural characteristics. First, growth rate factor, and second pigment production. In growth rate, see here first what is, that is rapid grower. Example Vibrio cholerae, and second slow grower, that is Mycobacterium tuberculosis. Next, pigment production. Pigment producer, it is Staphylococcus aureus, and pigment non producer, it is Escherichia coli. coli. Yes, this is all about the classification of bacteria or morphology of bacteria. Here we completed the structure, ultrastructure of bacteria and the class different ways of classification of bacteria. We will stop here. In next lecture we will see the next point. Okay.